This is AP Bio, Chapter 9. We're looking at patterns of inheritance or Mendel's genetics. This is our third and final video. We're looking at what happens when we go beyond just determining dominant and recessive patterns or inheritance patterns for traits based on meiosis and the passing on of alleles. And so if we look at, we're going to look at some of the different patterns or way traits are passed on in a family, because really we're looking at genetics as a way of looking at inheritance, and often when we think of inheritance, we think of generation after generation of individuals. And so it turns out that the inheritance of many of the human traits do follow Mendel's laws, the law of segregation, the law of dominant and recessive. And so if we were to look at several generations of a family, what we would do is we would draw something called a pedigree. And a pedigree can actually show individuals' genotypes based on their phenotypes or what they look like. So here we're looking at a pattern of deaf versus normal hearing in a family. And we are starting with the grandparents, which are all the way up here. Then we have parents. And then, of course, these parents were very busy making these children since they have so many of them. Now, what you'll notice about this is that some of the individuals are shaded in. Jonathan Lambert and then two of his offspring. If they are shaded in purple, what it means is that they are deaf. And if you look at their genotype, which is listed directly below them, you're going to notice that they're all homozygous recessive. In order to be deaf, you have to have inherited 2 recessive alleles from your parents. Now, if we look up higher here, let's start with Joshua Lambert and Abigail. What we notice about Josh and Abigail is both of them have normal hearing. Yet when they got together and they made an offspring, one of their offspring, which was Jonathan, turned out deaf. How did that happen? Well, it turned out that both parents are what we call carriers. Now, if we look at this Punnett square that we could have done for them, So what we notice about both of these parents is that dad was normal, and so here's our big D and our little d, and mom was normal, big D and little d. So when you do the Punnett square, you get big D, homozygous, dominant, you get heterozygous, possible offspring, and then lastly, you get the individual offspring that turns out homozygous recessive and has the deaf characteristic. So again, let's go back to our family pedigree. Here are our normal parents. Here they are, and they have a 25% chance of having made a homozygous recessive offspring, which was Jonathan, and he is deaf. Now, when we look at this kind of in bigger picture, we can ask, well, what does this mean, or how does this happen? Well, it turns out that mom and dad both carried the recessive allele for a trait that causes a genetic disorder. And because they both have the allele, but they don't look like it, we call them carriers. Carriers become very important as we start to look at and talk about what are the traits and how are they being passed on. So now let's look at some examples of some of the human genetic disorders that can be passed on. Here is some of the inherited human genetic disorders that are possible. When we look at them, we're saying that they are all autosomal. And autosomal means it's on any chromosome except X and Y, which are your sex chromosomes. So we have recessive disorders and we have dominant disorders. An example of a dominant disorder is dwarfism. In this one, if you inherit even one of the alleles, you will be a dwarf. So that would look like or have dwarfism, and so would homozygous dominant. Now, as we look at this, we're going to notice that some of the disorders are only possible when you have recessive, and so you have to have inherited both homozygous recessive alleles. And one of the examples of that is sickle cell anemia, and sickle cell anemia, here's an example of blood. So here you can see a normal blood cell, and then here you can see sickle cell. Now, if you inherit 2 alleles for sickle cell, all of your blood will look like this. And when that happens, you get a variety of health issues. However, if you're a carrier, so you have one dominant and one recessive, you'll have your blood will have some normal and some sickle cell. and you are known as, again, the carrier. And so you are healthy, you're fine, but you're able to pass on the sickle cell. And if the person you have children with also passes on sickle cell, then you'll create an offspring with the homozygous recessive inherited genetic disorder that has sickle cell and all of the health risks that are associated with it. If we're talking about carriers and passing on genetic traits, what's happening is that with the kind of inventions of new technologies, what we're able to do is we're able to look at our genetic legacy or look at our genes before we pass them on. And so we can have or kind of get reproductive decisions based on, are we a carrier? So one of the major things that happens when we do genetic testing or screening before somebody reproduces is we're just trying to identify what are the possible genetic disorders that you might carry, not have, but carry. And does your partner carry them as well? And then what are the probability that you would then create an offspring that has that genetic disorder? So one of the things that they do, or one of the ways they're doing this, is fetal testing. So what the doctors will do is they'll simply go in and take a small sampling of the amniotic fluid. And when they do that, they'll take the amniotic fluid, they'll spin it out, and then they'll look at a picture of the DNA. And the picture of the DNA is called a karyotype. And by looking at the karyotype, we can actually identify some genetic disorders. Some of the genetic disorders that are famous is Down syndrome, which is caused from trisomy 21. And what it means is that the 21st chromosome, rather than having just 2 copies of it, you get a third. And the third is what causes all of those signs and symptoms of Down syndrome. Another way that we're able to look at or kind of analyze for genetic defects is through fetal imaging. So if you go in and you're pregnant, what they'll do is they'll take a picture of the baby, and we can actually look at a sonogram or the image to see if there's any sort of problems. The last thing that happens is when a baby is born, we're doing newborn screening. And there's some genetic disorders that can be detected at birth. One of the best examples is PKU. Phenyleucoronea is detected by a simple blood test. However, if a child has PKU and it's unidentified, they can actually become mentally deformed because they've been exposed to certain phenylalanines or a product that's found in food and breast milk. If we know that the baby has PKU and we avoid phenyleucoronea for their phenylalanines for the entire adolescent life, the individual grows up to be perfectly healthy and normal. And so newborn screening has become increasingly common as we get more information about genetic defects. With the idea that we're looking at what traits can be passed on, up to this point we've simply said dominant to recessive. However, the relationship between genotype and phenotype is rarely that simple. And so oftentimes the genotype does not simply say, okay, it'll be this phenotype based on dominance, it'll be this phenotype based on recessive. And some of the examples are things like incomplete dominance. So certain traits, the offspring has a phenotype somewhere in between the two if it's heterozygous. So in our example here, we have a homozygous dominant red flower being crossed with a homozygous recessive white flower. And normally we would have said crossing red and white, dominant recessive, the offspring would all look red. They would all be heterozygous, big R, little r. Well, in some cases, the big R, little r heterozygous actually turns out pink, because when you combine red and white, you end up pink. And this is known as incomplete dominance. One of the more real-life examples of this, not just using flower color, is with low-density lipoprotein receptors in humans. A normal person has lots of LDL receptors, and so they take in cholesterol. Now, if you are homozygous recessive, you have no receptors, and so you have high cholesterol. And as we all know, high cholesterol creates health risks. However, if you're the heterozygous, rather than having the normal, what you look like is you have somewhere in the middle, between a lot and a none, you have a few. And so you have a mild case of the disease. Not only can we have incomplete dominance, but what we can have is something called codominance. Codominance is what happens when you have a offspring. So here we have our homozygous dominant. Notice that they have the white at the tips. If you look at our homozygous recessive, they have the white and the clover in the middle. Now, if you're a heterozygous, again, if we had simple dominance, you would have white tips. However, if you look, they actually have both. they have both traits, so it is co-dominant, kind of like co-pilots. So codominance is when you have both traits or phenotypes expressed in the heterozygous. Another example of some of the things that can happen beyond simple dominant and recessive occurs when we have more than one allele. So beyond the idea of incomplete and codominance, what can happen is some traits or some phenotypes actually exist in more than two alleles. And so there's often multiple alleles for a characteristic. And one of the examples is blood types in humans. We have 3 alleles. There is the A, the B, and the O. So again, there's 3 types. It's not just dominant and recessive. Now there's the A type, the B type, and the O type. The interesting thing about blood type is that A and B are co-dominant. So if you are AB, you will express both traits. Now, if we look down here at this chart, what we'll see is that the blood group O is simply recessive to everything. So O is recessive to A and B. But A and B are co-dominant. And so there's actually 3 different alleles, each with their own dominant, recessive, co-dominant patterns. Continuing with this idea of how do we make it even more complicated, another example that can happen is where you can have one gene affect multiple phenotypes. And so one of the best examples is going back to our sickle cell. So here's our sickle cells. Notice if you have sickle cell, what happens is you get lots and lots of different phenotypes. Looking at this chart, what you'll see is that you can have kidney failure, you can have rheumatism, which is like arthritis, you can have pneumonia, you can get paralysis, you can have impartial renal function, so your kidneys aren't quite working, and that's a result of having sickle cells, which causes the breakdown of your blood cells. It causes clumping and clots. It causes sickle cells to build up in the spleen, which is what filters out bad blood. And so all of these things happen as a result of having the one allele or homozygous recessive. With this being said, it's not always a negative thing to have multiple phenotypes or multiple results happen from a trait. And one of the best examples of having one trait, such as skin color, being influenced by multiple alleles is that it creates a wide variety of let's just say, skin tones or wide variety of phenotypes. And so oftentimes when you have this continuum of phenotypes, so you can have skin color from very light all the way to very dark, with all the shades in between, we know that it's caused by multiple genes. However, even with skin color, sometimes what happens is the environment affects it. So if any of you have ever been out during the warm summer days, what you'll find is that your skin tone will change. You can get tan. And so it turns out that not only are your phenotype based on your genes, but it's also based on the environment in which you're in. If we're talking about all of these traits, the genotypes and phenotypes, what we need to go back to and remember in our mind is that we're talking about Mendel's laws, so all of the traits that Gregor Mendel figured out, and what role this plays in just our overall DNA. So the genes we're talking about are located on chromosomes. Chromosomes are made up of our DNA, and it's passed from one generation to the next. How do we pass it? We're using sexual reproduction, so we're undergoing meiosis. Now, meiosis is what tells us what the inheritance pattern is going to be. So here we can see a base, kind of just a general example of meiosis and all the possible combinations that can be made from an individual's alleles. However, when we look at a chromosome, it's not just one gene per chromosome. And so we have to remember that genes that are on the same chromosome tend to be passed on together because you inherit a chromosome, not the gene. However, there is some complication that happens with this idea because there is some mix-up that happens during meiosis, and that's with crossing over. If we talk about genes being inherited together, what we're saying is that genes that are on the same chromosome, you inherit an entire chromosome. But as I alluded to, during meiosis, a process happens that kind of mixes up this process, and that is called crossing over. What happens during meiosis 1 is that the chromosomes hang out together so closely that they end up swapping genetic material, as you can see here. There's some genetic material that has been swapped. So what does this look like up close? So here we have a diagram. So here is our 2 chromosomes. They're hanging out. And what's going on is they have come together so closely that they will literally cross and swap material. So that what ultimately occurs is you're creating a chromosome that looks similar to those shown here. We have crossed the material. And then as we go through meiosis, what happens is that we ultimately end up producing daughter nuclei that are different. Each and every one of them is unique as a result of what's been going on. Now, with this, how did we figure out this crossing over was occurring? There's a new scientist called Thomas Morgan Hunt, and we'll talk about what he did with flies. So what Thomas Morgan Hunt did was he looked at fruit flies. And the fruit flies that Thomas Morgan Hunt was looking at were either gray-bodied with long wings, females, or they were black-bodied with short wings, and they were males. Now, when he allowed the gray-bodied long-winged female to mate with a black-bodied short-winged male, what happened is he found offspring of all 4 varieties. He found offspring that were gray with long, just like mom. He found black with short, which is just like dad. But then he found some that were different. He found some that were gray with short wings and some that were black with long wings. And he said, wait a minute, how does this happen? Well, by looking further at the process, what he was noticing is that the female had 2 alleles. And they were crossing when she was producing her gametes, her eggs. And by having that occur, it allowed for offspring to form that would have that crossed over or that different phenotype. And by looking at how often the crossing over occurs between 2 alleles, you can tell how close they are on a map. And so in short, the closer alleles are, the less likely crossing over is to occur between them. And we'll do some practice problems with this in class. Now, this is talking about genotypes, phenotypes, traits, passing them on. What we now want to talk about is the sex chromosomes and what role they play in genotype and phenotype. So, with sex chromosomes and genes that are found only on the sex chromosomes, what we're talking about is in humans, the males have an X and a Y, and females have 2 X's. The sex-linked chromosomes sex-linked traits are often on the X chromosome. So males only have 1 version of it, females have 2 versions. Because of that, when we talk about becoming a male, what ultimately is being said is that if you have a Y chromosome, you then contain the genes for the development of the testes or the male reproductive system. But if you don't have that Y chromosome, you will therefore develop into ovaries. So females are the default sex for humans based on our sex chromosomes. However, other species of animals have different patterns of sex determination. So for example, in crickets, females have 2 X chromosomes, but males only have 1. 1 X chromosome says you'll be a male, and you have no other chromosome. And if you're a chicken, if you're a male and a female, you actually have different chromosomes. So the males have 2 Z's, whereas the females have the W. And then in honeybees, it's actually a difference in the amount of chromosomes. females have 32 chromosomes and males have half that number at 16. Now, the reason why this matters is because sex-linked traits are then more likely to be passed on in males. And the reason is that they only have one version of a gene. Because of the inheritance patterns for males only having one X, what happens is that any sex-linked trait gets kind of a unique pattern. And what happens is that it's more likely to show up in males. So let's talk about some of the examples. With Morgan Hunt and his fruit flies, what he found out or discovered is that in Drosophila, white eye color is actually determined by what sex you are. Males are more likely to be white-eyed. And if we look at a Punnett square, we can actually see why it's more likely to show up in males. So here we have a female who is homozygous dominant, and we have a male who is recessive. Now, if a male passes on his X chromosome, what happens is that he's passing on a recessive trait. And so it's not going to show up because the female will donate either the dominant or the dominant. That's all she can donate. So you're going to create a offspring that is red-eyed or dominant, no matter what happens. However, if you look at a male and a female, and the female is a carrier of the white-eyed or recessive trait, but the male has the dominant, what happens is that when you cross them, you only have a 25% chance of creating a male with that recessive white-eye trait, but it's impossible to create a female because the female will always inherit a dominant X from the father. Now, if the male is white-eyed and he crosses with a female who is a carrier, so she's heterozygous, what happens is you have a 50% chance of making it. You're likely to make a male with white eyes, but you have a small chance of creating a female. However, she has to inherit both the recessive X from the mom and the recessive X from the dad. So with that being said, and looking at all the different possibilities for Punnett squares, what we now know is that sex-linked disorders most often affect males in the inheritance pattern because a male only inherits the one X. And so if he inherits a recessive X, then he has the trait. Whereas in order to create a female with the recessive trait, she has to inherit both recessive traits, and the father has to have the trait. One of the best examples is hemophilia in the royal family, looking back at history. We'll look at this in more detail within the class lecture. And so if we want to summarize everything we've kind of looked at so far for Gregor Mendel is, we know that genes are dominant recessive. We now know that they can be codominant, where you see both traits in the heterozygous, or they can be incompletely dominant, where you see something that's in the middle of the two traits in the heterozygous, or they can be sex-linked, and the sex-linked traits are those that are found on the X chromosome. Because it's on the X chromosome and males only inherit the one X from the mother, it's more likely that they will show up in, or sex-linked traits will show up in a male generation. And we do know that all individuals have two alleles for a trait. The exceptions to the rules are those traits that are polygenic, where you have more than one gene controlling a trait, such as skin color, or you have multiple alleles for a trait, such as blood type, where you can have an A, a B, and the O. This is our last slide for our lectures on genetics and the patterns of inheritance. We'll continue to discuss this in more detail as we get into each of these examples within the class.